Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Stephen Anderson. He is an associate professor in the Department of Molecular Biology and Biochemistry at the Rutgers New Brunswick School of Arts and Sciences. Welcome, Steve.
Hi there, Marty.
Good.
Thank you for setting this up.
Okay, it's my pleasure. So, Steve, let's begin in the beginning. Can you tell me a little about your early education and what you were doing before you came to CABM?
Well, I grew up in Nebraska, and both my parents went to University of Nebraska, but my family moved to Washington D.C. area when I was a preteen, and it opened my eyes to a lot of new things. And I ended up going to Harvard College for undergrad. And interestingly, I'm old enough that I could major in biochemistry, but there were no undergraduate biochemistry courses. So, and partly the reason I did that was that I could take any science course I wanted and have it count towards the major. And then I actually just carried this over into grad school. I stayed at Harvard and got my PhD there. Then after that, I wanted to, I was trying to cite on a postdoc, and I'd done all this incredibly quantitative work as a grad student, and I wanted to get away from that, but kind of not to get too far away. So I went to the Sanger lab, and that was fantastic. Actually, it's hard to describe, but what they were working on then was sequencing the 2 mammalian mitochondrial genomes at the time, which was the human and the bovine mitochondrial genome. And the third one was going on at Stanford on the mouse mitochondrial genome. And Fred was also sequencing lambdaphage. And at the time, we figured out more than half of all the DNA sequencing in the world was going on in the lab. And I was the only postdoc. So then my next step was a little complicated. I had offers from some universities like UCSD, but I decided to go to Genentech as my first job after my postdoc, which raised some eyebrows, I'll tell you. So I went to Genentech, and I actually wrote an NSF grant, became an associate, adjunct associate professor at UCSF. So in the end, I had grad students and postdocs.
Can you tell us when you came to CABM and what brought you here?
Well, I came to CABM 30 years ago. Roughly, I was in the class, as it were, with Celine and Peter Lobel. And so that was in end of 1988. And basically the reason I came to CABM was because I had a very positive experience for 10 years as a postdoc in England and at Genentech in San Francisco. And I found the faculty and staff at Rutgers, UMD&J, and CABM to be very open and smart and collaborative and collegial. And it was just, it was just fun to be with them as colleagues. And that's important for science. And the second reason was I'd always wanted to live in the New York, New Jersey area.
Sure, real life intervenes. What's one project you're working on now that's particularly exciting to you?
Well, I'm glad you asked me that, because I'm really excited about it. And that's because my background in structural biology and protein folding has intersected with some recent research which was funded in our lab involving antibody generation, antibody generation against human proteins. And what we do is we engineer them to change their conformational dynamics. And then we create cocktails. So we're sort of making a super antigen that's a cocktail of multiple antigens.
Now, I know you're doing some work on COVID-19. Do you want to tell us about that too? Yeah,
well, thanks. Thank you for asking. And that's partly due to you, because early last year when this happened, we had a faculty meeting and you said, well, anybody that's doing anything that could be could help, please, please consider, changing what you're doing and applying it to the COVID-19 problem. and we started working on a vaccine for COVID. And we did something a little differently because in the early days, people were so afraid that it may not be easy to get a vaccine that they made these very large, very fancy vaccines like the Pfizer vaccine and the Moderna vaccine. And it turns out it's actually not that hard to make a vaccine against COVID that works. Most of the vaccines that are being developed work. And so it was fortunate because we decided to make, can we make the cheapest, most easy to distribute, easy to administer vaccine in the world without sacrificing safety or quality. So that's what we've been aiming
Up
at.
there, because I don't want you giving away too many industrial secrets,
No.
because you need to apply for a patent on this stuff.
Yeah.
So
Well, I have.
good. That's good.
Senator,
Have they filed yet? Have they
Yeah,
filed it?
filed, yeah.
Okay, good. So then, all right. But it sounds very exciting to me, and because there are, it's exciting to me because there are 8 billion people in the world, and they're not going to be able to afford the Pfizer or the Moderna vaccine. So having some alternatives, especially very high titer, may be important, especially if the virus starts changing a lot.
Yeah, and this technology we're developing increases diversity. So it may enable the vaccine to, even if the virus starts to drift,
Good.
it could encompass those.
Yeah. Steve, how do you interact with students and trainees in your lab?
Well, in the 90s, I had a lot of undergrads in the lab doing research, and then also in the early 2000s, and there was a very large number when I was involved with the Northeast Structural Genomics Consortium with Guy Monteleone. But now I teach pretty intensively. I teach 2 undergraduate courses, one each semester, and there's a lot of fantastic students in those courses, and I'm often asked to be on their committees. So rather than, once in a while I'll supervise in our own lab somebody doing a thesis, but mostly interacting with them.
Great. So let me ask you my final question, which is it's a little bit of a weird question, and that is, imagine that we were writing your scientific obituary. And I wanted to ask you, how would you like to be remembered as a scientist? What would you like to be remembered for?
I think this goes back to Sanger. I think I sort of saw him as almost a Galileo-like scientist in the sense that he was inventing not a telescope, but a new techniques, sequencing both proteins, RNA, and DNA. And
yeah, for those who don't know, Sanger didn't just win 1 Nobel Prize; he won 2 Nobel Prizes. So.
Right, and actually, I was in the lab the day he won the 2nd Nobel Prize, which is something that's not to be missed. If you, I mean,
yeah,
I don't
not
want
a very common occurrence.
I mean, it's one of those days in your life. But anyway, so I thought, looking at Fred and admiring him, that I really resonated because I think if I had to be remembered as somebody, I think it's that I enjoy making tools and technologies that can be used to push back, employed by others to sort of open up new areas or push back the boundaries of ignorance. And so that's as an epitaph.
That's good epitaph. That's better than most. Fine, Steve. Well, I think you've fulfilled your obligations in this interview. I think it's going to be very interesting for people to watch. So thank you very much.